Thank you, Alisa. And the floor is yours.
Okay, perfect. Thank you. Thank you for the introduction, and thank you for the opportunity to come and talk to you a little bit about what we've been doing in my group. I appreciate the opportunity to come to speak, as well as for all the talks that have come before me. I think that what we do kind of bridges a lot of what's been talked about already, and especially thank you to Joe for that introduction to metagenomics and to antibiotic resistance, and personally for welcoming me into your group, what feels like all those years ago, and getting me started on my path here. And so I'll show this this overview of functional metagenomics. I think we've all got some sort of slide like this where we can walk through, and I won't walk through since I think we're all pretty familiar with this, but there are some things that have started to stick out to me about this process that have gotten me thinking a little bit. And mostly we're kind of down here at the bottom, where we're going to go through, we're going to start with some sort of sample of interest, and we're going to go through and make a metagenomic library. And right here, to me, I've always thought about this as an or moment. We're either going to do some sort of functional screening, or we're going to do some sequence-based screening. And we've seen a lot of nice examples of the power of taking both of those approaches already here today. And what I will hopefully convince you of by the end of the talk is that if we think about this as an and approach and not an or approach, we can really leverage some of the power of both of those technologies. And that's really what I want to think about there. The other thing that really stands out to me about what we've got on this slide is really what the end goal is. And for me, I would always say that the end goal is I want to find something novel, right? I'm looking for a novel enzyme. I'm trained as a biochemist. I want to find something novel. And more I've been thinking about, what does that actually mean? What kind of novelty am I looking at? And hopefully in the next few slides here, we can walk through some of the ideas that my group has had about what is novel and what kinds of things are we looking for here. So I'll move on and show you. This is, don't worry, don't strain your eyes trying to read this. This is an overview of the results of a recent selection that my group has done. So these are data that we published recently, where we went to a wastewater treatment facility and we asked what kinds of antibiotic resistance genes are in that treatment facility. And we used the technology pretty much identical to what Joe described this morning. We made phosmid libraries, we selected an E. coli, we looked for clones that grew on different antibiotics. And so here's what we get. And I've got a couple highlighted in red because they highlight a few different aspects of novelty that we were looking for. So the first one, and I'll jump to the bottom here, this TET62. This gave growth on tetracycline. When we looked at the active gene there and the product itself, it was 68% identical to the closest known homolog in the database, which the tetracycline community is a little bit different than some of the other antibiotic resistance communities. There's a very strict threshold of what you can call novel. 79% is the threshold, and we were under 79%, so we applied to the community and we were given TET62. That is the name of our novel enzyme. So it's a little more straightforward in that case. We've got something that the community has agreed upon is novel enough to get a new name. Some of the others are not so clear. So up above, I've got this where the clone is called spec B. If we look, it's 57% identical to the closest known relative. Is it really novel? I would hope so. It was interesting enough for us to follow up on and to try to figure out what's going on there. So it's predicted to encode this aminoglycoside nucleotidal transferase. Spectinomycin is an aminoglycoside antibiotic, so that is not all that surprising. And just kind of looking at what we have, if we put this into E. coli, we get about a 5-fold increase in antibiotic resistance over our control. Again, it's predicted to encode this nucleotidal transferase, pretty low identity, about 57%. But then if we go ahead and we line it up with the closest known homolog, and we start to ask the question, where is that sequence homology housed? And so what I've got denoted here, my query line is the clone we found, the closest known subject in the database is down below. The things denoted with red asterisks are those amino acid residues where we've got substrate or cofactor binding, the things involved in the biochemistry. And what you can see is that we're actually fairly well conserved in those regions. So while we've got overall a little bit lower sequence identity, perhaps there's some novelty there. If I think about it from a biochemical perspective, I don't know that we're catalyzing any new chemistry there. And so I'll go back to our overall results and see, okay, so we've got a couple of examples of things that are maybe novel in different ways. We've got that TET62, we've got spec B. What about this one I've got? I've got spec C here. Spec C is 100% identical to a known aminoglycoside resistance gene. It's known it's identical to this known streptomycin, spectinomycin adenyltransferase. Well, since we did this through a functional assay, this was a phosmid clone, we went back and asked, what about the flanking DNA? What about the genetic context where we find this? And that's where we went ahead and we looked and we found that this is on a class 1 integron. And class 1 integrons have a specific architecture. They've got this int I1 gene, that's the integrase. Oftentimes, they'll have this 3' consensus sequence, although not always. And so we found both of those, and what's in between is the variable region. And that's where we're going to get different antibiotic resistance genes, different cassettes that have gotten integrated into the integron. What I've got here as the black boxes, those are the integration sites where the integrase might insert new cassettes, new antibiotic resistance genes. And so we went through and we looked at this clone, the one where I've got 2 arrows right here, that was that spectinomycin adenyltransferase that we identified. But if we look at the different resistance genes around it, we've got a beta-lactamase, we've got something that's predictive for lincosamide resistance, we've got a different aminoglycoside resistance gene, we've got some quaternary ammonium salt resistance here. And so there are databases of integrons that we could go through, and we went ahead and we compared this to the integral database. And this was different enough. They had not seen any like this. So this arrangement of cassettes was different so that they gave us the name of IN 1875. So again, a different aspect of novelty here, right? In this case, the gene itself that we found that gave the antibiotic resistance was not novel, but the genetic context was novel for us. A couple other interesting factors that came out of this one. You can see there are 2 copies of the active gene. We did PacBio sequencing with long read technology to make sure this just wasn't an assembly error, and it's not. It really is duplicated on the integron. Some examples of that in the literature, where we know that sometimes we can get resistance genes that are duplicated. The other thing is this lincosamide and this, it's a nucleotide phosphorylase, seem to be transferred as a single cassette. There's no intervening little black box in there. So those seem to be transferred as a single cassette when they move. So again, things that were a little interesting to us. But as I come back to our overall goal and our table here of our results, we do have some aspects of novelty, I would say, to the things that we found, right? And certainly when I'm going to publish it, these are novel, right? But when I set out to find a new resistance gene, for me, kind of that holy grail is I want something that doesn't look like anything else, right? Every one of these genes that we found on every one of these clones, if we go through and blast it against the database, it would be predicted to be an antibiotic resistance gene, right? It's got that function that we know. I want to try to move beyond that. How do we get to something that from sequence alone, we would not predict to be involved in antibiotic resistance? That's really what I want to move towards. And that's what we're going to talk about for the second part of the talk, is that we were able to do that. And so we'll talk about how we did that and how we kind of leveraged the different technologies to move towards that different definition of novelty. The other thing, I show this just because some of my students might be on the Zoom meeting. I feel your pain. This was a very low throughput kind of thing. So in order to generate the data on this table here, I had multiple students working in a lab over the course of about 3 years. And they ran hundreds of gels that looked like this, whereby we were going through clone by clone and trying to find those unique clones that we would follow up on. And this is just, this is a representative gel here where we're going through, and each lane is a potential clone and an RFLP analysis just to weed out the different phosmids that have the different banding patterns. And so I've got the banding patterns kind of labeled here. But again, my students, they spent years doing this to sift out hundreds of clones down to a table that looks something like this. So again, very labor intensive process. I get it that nowadays we could go through and we could do, a more high throughput sequencing analysis, which has some advantages and some drawbacks that we can talk about a little bit later if that's what we'd like to do. But I started to look for other data sets and other ways that we could go ahead and try to find this new novelty. And that's where I came upon this study here, whereby they went ahead. This is a group at the FDA that went ahead and they were interested in surveillance. And they wanted to surveil for potential pathogens and antibiotic resistance genes. And what they did was they went around to retail turkey samples, isolated salmonella bacteria, and cultured them. And they asked questions about whether they were resistant to antibiotics, but then they also sequenced every one of those genomes. And as I start to think about doing a little more sequence analysis, I'm at a pretty small university. We don't have a genomic center. Access to a public database like this is crucial to what we were doing. And so we wanted to take a look at what they found here. And so in here, we would get a table like this, whereby each of the isolates was classified as to their genotype, whether they're genotypically resistant or susceptible to gentamicin or streptomycin or a whole host of antibiotics. And so those data are there and they're publicly available. But then they took it a step further and they said, let's layer the phenotypic data on top of that. And so now if we classify these isolates as either phenotypically resistant or phenotypically sensitive, we can go ahead and compare that to the genomic data. And we can see overall, they're pretty good, right? They just took their genomes, they ran them through known databases like CARD or AMR Finder or something like that. And you can see that, in the group that's phenotypically resistant, out of 105 that were phenotypically resistant, they were able to predict 99 of those just based on known genotypes. Or the ones that are phenotypically susceptible, out of 535, they were able to predict 530. From a surveillance perspective, that's not bad. They can do pretty good. They're upwards of 90% on all of these. But from a discovery perspective, I'm looking at the other columns. I'm looking right here. There were 6 isolates that were phenotypically resistant, but had no known recognizable gentamicin resistance gene. To me, that points at, please don't tell me time yet. To me, that points at the opportunity for discovery, right? That's where I want to be. That's where I was directing my group. These have things where when we have that genomic data, we have no idea, we're not able to recognize the resistance gene that we know is there because they're phenotypically resistant. And that's where we were headed, and that's where we were going to start to mine. A different overview of this one, you start with your favorite sample, right? It's always that one troublemaker chicken. We go ahead, we isolate the bacteria that we're interested in, and we start to ask, phenotypically, each one of those isolates, is it resistant or susceptible? And we compare it to the genotypic prediction, resistant or susceptible. And right here is the one that we're interested in. Things that are phenotypically resistant, but have no recognizable gene when we compare it to the database, and we're calling these GSPRs, genotype sensitive, phenotype resistant. So for us, my group is really good at doing functional metagenomic screens. We call the FDA, we ask for these isolates. These are going to be the basis of the library we create for a functional screen. That said, I'm impatient. It takes a while to call the FDA and get your samples, and there's paperwork to do. Let's see if we can take it one step further. Again, we have the genotypic data, right? The genomes are sequenced, and the answer is in there if we just know how to ask the question. So while we're waiting for the isolates to arrive, we go ahead and we ask the question, can we computationally predict that unknown resistance gene that doesn't look like any other resistance gene that we're about to pull out of our functional screen? And that's what we set off to do. My students are courageous and maybe naive. When I challenged them with that, they said, sure, we could do that, right? So this is the thought. And this is, again, just a different view of what we were looking at. I've got highlighted here, if I think about those isolates and I think about their genomes are a collection of genes. We should be able to line these up. And the ones that are GSPRs should have a gene that none of the others have. Computationally, we should be able to pull that out. And again, when we think about being naive, I am naive. I am not a bioinformatician. To me, that sounds like a computational problem we should be able to solve, but the we that's going to solve that is not me sitting at a computer. So I rely on the expertise of my students. I hired some really talented students that are interested in data analysis that were able to do that. And so that's the idea, is that in this simplistic view, we should be able to pick out that novel antibiotic resistance gene that doesn't look like anything else from the data set. And so getting back, this is the idea, is that we'll go through our normal functional selection. Once those isolates arrive in the mail, we'll do our functional selection. We'll arrive at that antibiotic resistance gene. But while we're waiting, maybe we could do the computational work and predict that same gene, right? And that's the goal. And overall, I think that that's going to be a really important tool for us to be able to use, because in this case, I'm talking about salmonella isolates that we can get from the FDA. What if we were talking about pathogens or bacteria that we couldn't get? What if we could go to these data sets and computationally start to predict these unknown genes that don't look like any other things in the BLAST databases? That's what we really want to work towards. So for us, it's a little bit of validation of our methodology as well, right? So hopefully we have a convergent workflow here where our functional selection is going to lead us towards the same gene that our computational prediction is going to pull out. So that's what we set out to do. Again, getting back to that table, the thought was, what if we can go ahead and look and compare these 6 isolates right here? And we started with gentamicin, quite frankly, because it was at the top of the table. So we start with gentamicin. We've got 6 isolates that are GSPRs. Let's compare those to the 530 that are genotypically sensitive, but also phenotypically sensitive, right? They shouldn't have any novel gene, any novel gentamicin resistance gene in them. And we go through and we run our pangenome analysis. So we run our pangenome analysis, we compare these sets, and the output we got is that it jumped right out at us. So we found that there were 2 genes that were common to these 6, but did not appear in the other 530 in the data set. And the other numbers here just tell us that, yes, they're in all 6 isolates. The genome fragment, the number doesn't matter. What matters is that they're the same genome fragment. So they're found on the same fragment of the genome, and they're adjacent to each other. They're genes 1 and 2. And this is where we can kind of go back and forth and start to do our functional analysis and start to look a little more at the computational analysis as well. We went back to those assemblies, those published assemblies, and said, let's go find those contigs. Let's see these 2 genes that are adjacent to each other. Where are they in that genomic data? What does it look like? And what it turned out is they were on very small contigs that only contained these 2 genes, which immediately made me question the results. Right? They're necessarily near the end of a contig. Lots of different assembly errors could have happened. There's certainly a lot of validation we need to go through to verify this. That said, it was hard not to get excited about it because I was pretty sure that the transposase was not giving antibiotic resistance, and it really was this hypothetical protein, this protein that nobody had characterized yet. And so I tried to contain the enthusiasm and maintain my scientific skepticism and move on. So at this point, the isolates are on their way. They're in the mail. We're about to do our functional metagenomic selection, right? And what we do for functional metagenomic selection is we're going to go ahead, we're going to isolate the DNA, we're going to clone it, introduce it into our surrogate host. Well, while we're waiting, the other thought we had was antibiotic resistance genes are often on plasmids. What if this new gene is on a plasmid? We could go through and figure that out. So we went through, these are the different isolates we were looking at that we had asked the FDA to send. We went to the genomic data they had published. We ran it through Plasmid Finder, and sure enough, most of them have plasmids. And so the thought was, as soon as the isolates arrived, we would just do a direct transformation. Before we worry about cloning a metagenomic library, let's just take a little bit of the genomic DNA, directly transform E. coli, plate on gentamicin, see what grows. And sure enough, there was a natural plasmid in there, a natural plasmid that contained our resistance gene and gave gentamicin resistance. So we were able to bypass the cloning step of the metagenomic, of the functional metagenomic selection, which was certainly helped our efficiency. And so we found this plasmid that we're calling PZJ18. It's rather small, it's only 9 kbs, and it's got an interesting architecture. One half of the plasmid, kind of in this bracket here, has a pretty common sequence that has been found in Klebsiella and other Salmonella isolates before. The right half contains some sequence that's been found in Salmonella genomic data as well, including ours. And if we look at what was on that plasmid, the 2 genes that we predicted were right there in the middle. So our computational prediction said that we should have a hypothetical protein and a transposase. I've already got the answer kind of drawn in there. That's my hypothetical protein, and there's my transposase. And so we went ahead and we named that hypothetical protein GRDA for gentamicin resistance determinant A. So we found this novel resistance gene. We found this gene that before was a hypothetical protein, and now we're able to give it this function. We also went ahead and we were interested in the architecture of this plasmid. So we went back to the original data set, right? And again, you can see I'm kind of jumping back and forth between our genomic data and our functional data. Now we've got the functional data. We've done our transformation. We found our clone. Let's go back to the genomic data and see what's in there. And we did find, we found lots of isolates that had this part of the plasmid that doesn't contain the resistance gene, but did not have the resistance gene. And sure enough, those were susceptible isolates in the original analysis. We also found multiple isolates that had the GRDA part of things, but not the other part of the plasmid, which was interesting because it told us that maybe we don't necessarily find this on this particular plasmid. Maybe it's in different genomic context in a lot of the different isolates, which again was interesting to us when we start to think about the molecular ecology of how this might be spreading, right? We've already done the experiment where this was found in Salmonella and we moved it into E. coli and show that it's functional, right? So it has the ability to be transferred. But now we're starting to find out, does it live on a plasmid? Does it live on a different plasmid or perhaps the chromosome? The only thing I can really kind of definitively say about that is that it's not always on this plasmid. So we're learning something about the spread of what might be going on here. We, of course, wanted to characterize spectrum of resistance. So we took our E. coli cells that have either the natural plasmid, or we went ahead and just cloned the gene by itself to make sure that sure enough, we've got the right gene on the plasmid. And we tried to see if it conferred resistance to other aminoglycoside antibiotics. And we can see we get resistance to gentamicin and cistamicin, but not the other aminoglycoside antibiotics. So we're learning something a little bit about the spectrum of resistance there. But again, I'm a biochemist. I want to start to think about this enzyme. It's good that we can follow this resistance gene. We learn about the molecular ecology, but I'm interested in the enzymology. What is actually encoded here? So one of the things we wanted to do is make sure this is really the GRDA gene and not the transposase that's active. So we use transposon mutagenesis to knock out the active gene. And sure enough, these transposons all hit GRDA. They didn't hit the transposase, which was reassuring. One of them fell right here. It's between the predicted promoter and the start codon. So presumably it's knocking out expression of GRDA, but the others actually hit the coding sequence. But now I wanted to zoom in a little bit more. And if I look at the GRDA is a relatively small gene, the protein that it encodes is only 167 amino acid residues. But if we look at those first 130, one of the things that we wanted to do was start to mutate it. And let's see functionally what's important to this enzyme. And this is where we actually did this in the course I teach. So I teach an undergraduate biochemistry course. Rather than giving the students a project with a known outcome, I gave them this gene and said, let's make some random mutants. Let's see if you can go ahead and find the functionally important parts of this protein. All we could tell from bioinformatics is that it's got a Walker A domain and a DXD domain. And those are both involved in nucleotide binding, so ATP binding probably. If we look at other gentamicin inactivating enzymes, oftentimes they'll bind ATP and use it as a substrate. And so this seemed to fit. If we look down below, any place that there's red, that's a mutant that my students in my course identified as part of their coursework. And we can see they cluster a bit around those functional domains. And then there's another hotspot out here that we still haven't figured out quite what's going on with that yet. So that's current work. That's what we're working on at this point, is can we purify this enzyme? Can we functionally assay this enzyme? Let's see what it's doing. And let's start to go through and look at these things like AlphaFold. Is this really folding up into something functional? Is this close to where this other domain is? Things like that. So I can summarize right there that, I think functional selections, we can go ahead, we can yield novel clones. We found, in that beginning wastewater project, different aspects of novelty there. But I think we also have gone ahead and we've been able to leverage some of the sequence data that's publicly available. And we can use that to maximize our efficiency. I can say for us, that original wastewater study took multiple students about 3 years to go through and sort through all those clones. Once those isolates from the FDA arrived on campus, we had GRDA cloned and knocked out of our clone in 3 weeks. And so that was quite the increase in efficiency for us. And that worked out really well for us. So with that, I will acknowledge, certainly the inherent acknowledgement here is to Joe for getting me started on this. But if I'm talking about just these, the projects we talked about today, the wastewater studies, I collaborate with a civil engineer at the University of Minnesota. So Tim LaPera. Shawa is the scientist at FDA who sent us the samples. I've gotten funded over the years from Research Corps and the Minnesota Environment and Natural Resources Trust Fund. So they make it possible. And then of course, the students. University of St. Thomas is a PUI. I don't have graduate students. So everything I've talked about so far was done by undergraduate researchers that have been involved in doing this. So if anybody wants to chat about that, I'm happy to talk with you more about that. But I am blessed with lots of undergraduate researchers that are fantastic to work with. I'll point out a few. So Bridget right here, Bridget led the wastewater project. She is, I'm told, months away from being Dr. Bridget. She's at Colorado State, so she's finishing up her PhD. Daphne Welter helped us out with learning how to do pangenome analysis. She left my lab and went on to Max Planck in Tübingen, and she is now at EMBL. So she's a staff scientist there. Ricky's a data analytics and computer science double major who I recruited into my lab. He's headed off to industry. He just graduated. Sydney and Happy contributed a lot to that GRDA study. Sydney is off to medical school, I'm told, next fall, and Happy went off to industry. And with that, I will wrap up and take questions.
Thank you. We have 2 questions of Joe. Maybe we could start with these.
Sure. Joe, after it's incubated, okay. Do you know if gentamicin is still present after incubate? Joe, if that's the question, right? Okay, so the part that I did not touch on. So do we know if the gentamicin is still present? I have a student working on that right now. So I've got a student. What we've been doing is we've been doing mass spec analysis of the culture medium afterwards. And I will say that we can detect, so the gentamicin that we use as a media additive has 3 different modifications on the gentamicin. We can see all those in the mass spec. It was a really tantalizing contaminant that we saw after GRDA. Something looked like it grew by about 100 mass units. I think it was 98 exactly, which looked really close to a phosphate. although my mass spec person in my department told me 96 is a phosphate, and we found that in the control sample as well. So that's definitely not the metabolite. But what we're working on right now is we're using a solid phase extractor to pull out the metabolite and concentrate it before it goes in the mass spec. And we're wondering if maybe we've got to tweak that extraction a little bit to see if we can actually get the metabolite. But that is very much, I was hoping to have those data for today. The student working on it graduated on Saturday, and so he's handed off to another student. I hope we have that. I'll say momentarily if they're listening, but anyway.
Thanks, Justin. Exciting stuff. With the GRDA sequence, when you threaded it, did you get any model of any significance that might tell you which group structures it fell into?
Tentatively. So we threaded it through, and I will say that when this manuscript was under review, somebody else found a homolog with similar active sites, similar size and everything like that, that they called GAR. So out of the Larson group, and they went through and they threaded through iTester as well. They found that kind of thing. Tentatively, so what they were saying is it looks like it might be a phosphotransferase. I don't know at this point whether it really is a phosphotransferase, or possibility is that it could be a nucleotidal transferase. I don't quite know yet. So those are still really preliminary, and we're trying to work through those.
Thank you. Any other questions for Justin? I have one for you.
Yes,
Thank you for presenting this so smart approach. So you detected additional genes in your pangenome analysis. I was wondering if it could be interesting to try detecting single point mutations in genes that are conserved in the pangenome.
Sure. Yeah, no, absolutely. Right. So can we pull those kinds of things out? Not being a bioinformatician and being naive about these things, I would say, sure, we should be able to do that. But we haven't tried it. To be honest, this was our first, okay, low-hanging fruit. Can we do this? Can we pull out a whole gene? Now, can we go back and do this from more of a, a polymorphism analysis? I think that would be interesting to do. I think it'll pose a little bit different challenge, but I think it would be interesting to try. So it's certainly on our list of things to try as a follow-up.
I have no idea of the promiscuity of such genes, antibiotic-resistant genes, towards different classes of antibiotics. Are there any data on that could be analyzed using AlphaFold and maybe targeting some particular residue that could explain the promiscuity and the resistance to different classes?
Sure. Yeah, no, absolutely. Right. And again, this is something that Joe touched on a little bit this morning, that, oftentimes substrate specificity is a little more loose with these, right? And so, yes, looking at the active site residues is very much where we want to do. And let's see, if we go ahead and do either computational docking in that active site, or actually, I would love to have mutants where we change the substrate specificity. That was part of what my students were doing. They plated for gentamicin knockouts, but they also plated on things like kanamycin to see if we could get a substrate specificity mutant, and we did not pull any out. So negative result doesn't tell me anything, but if we had pulled one out, sure, it would point to that kind of thing. And I'm very much interested in the biochemistry of what's going to dictate that. Absolutely.
With your libraries of mutants, it could be very interesting too.
Absolutely.
By using microfluidics.
There you go.
Okay. Any other questions? Yeah. No? So thank you, Justin, again.
Thank you, everyone.
We will continue. We're going to continue this session with 2 short talks. The next one will be by Jiang and Guiyin. Yes, it's you? No? No, in the program, I have in the last version of the program, no? Okay, yes. Are you ready or not? Yeah. Okay, thank you. Thank you. Jackson is a research scientist at SINTEF in Norway, and he's an expert of metagenome mining to discover enzymes for industrial biotechnologies.
Okay.
Thank you.
Yes, so in the next 10 minutes, I will try to give you an overview of some research activities at Syntef that involve genome mining and also functional screening for different for finding a novel answer. How does it work? All right. It's a laser. Okay. So we started our activity in genome mining a couple of years ago, and it only started with this Oxymorph project that's funded by Research Council of Norway. So basically, in the project, we want to look for new enzymes that can degrade lignocellulose materials. So it is a project with many partners, and we work with mostly with peroxidases and lacases, but we also tasked with the task to find new LBMO and other type of enzymes. So we look into our own actinobacteria strain collection and also some other samples that we have been collecting throughout the years. For example, there is some soil samples from Svalbard. This is samples of biofilm from the water treatment. From the oil reservoir metagenomics libraries. So also we also have some other samples from different projects like microbiome associated with microalgae. In the metafluids project, so we also have this the samples from the gut of this reindeer. So we have used different libraries to screen for the new enzymes. So basically we were a little bit naive also at the beginning. So we will start very simple first. So we use Hammer and Profile HMM to look for the new enzymes. So at the beginning, so we use this DBCon Profile HMM libraries because it's pretty very good K-SAMS HMM that we use for looking for carbohydrate active enzymes. So the target enzyme is LPMO, multi-couple oxidase or lacases, and peroxidase and other glucose dehydrogenases. So the workflow is more or less the same with other groups. So we start with the data set. So we use HM scan. Basically, we base our workflow also from the workflow that was developed from the group of DBCon. And we have the raw output from the HMM. So later we trying now to use ECNOG mapper also to do the annotation. So we have the raw output and we have some strip to take out the whole open reading frame, and we clean it up and we import into the database. And then we will use different approaches to find a short list, because usually you end up with a lot of hits and we need to have to go into the short list of the candidate that we come with later. All right. So this, for example, this is the K-SAMS, the abundance hit that we found in our actinobacteria strain collection. So basically, when we got the HMM hit, the question is like, okay, how we can get the sequence out? And at the beginning, we're also interested in the multi-domain enzymes. So basically, our approach is that, okay, this is the HMM hit. We have the coordination of the hit. So we walk back upstream until we meet the first stop codon, and then we walk downstream until we meet the last codon, and then the first stop codon. And then we take out the whole thing, and then to analyze what is the hit about. So at the beginning, I didn't know like what is the best approach to select the shortlist. So I use multiple sequence alignment at the beginning. So basically just make a very big multiple sequence alignment of all the hits. And then basically selecting what I call representatives of each of the cluster here. And later, after the talk of Ben from Arctic SAMS, he presented the approach called sequence symmetry network. And we use it more and more often. So we basically create the network of the hit, combine with the reference enzymes if it's available, and then try to select some cluster here or some single tone here, and then look into whether it is a new enzymes or not. And then later, of course, is the manual curation of the larger list into the short list. So we basically blast into UniProt, BDB, and CBI, just to have the evaluation like how novel is those sequence are. So so in the Oxymore project, so we able to find a new LPMO. So it's a multi-domain enzymes with from AA10 families with the CBM behind that. Also, we found a new decolorizing peroxidases, and surprisingly, we find a new conserved motif. So the deep time peroxidases have been reported so far. They have the GXXXDG motif. So for some reason, we found one that we have the EXXDG motif, and apparently it's still active. So we're able to find a new one, and then also we have the new thermostable small lactase, and with the lignin degradation activity, and with the Slack one here. So we built our own profile HMM based on the public database. All right, so that is the Oxymore project. So in other projects called MarBiotech, and later in the SFE industrial biotechnology. So we were looking for the plastic degradation enzymes and also the DNA acting enzymes. So we use the same approach from the Oxymore project, and then we will look for PET esterase basically, also using the sequence symmetry network and also multiple sequence alignment. Also, we combine it with functional screening. So, for example, in this case, is functional for polycaprolactone screening. So basically, we look for the halo, and later we adopt it into the hydroput method to look for the polycaprolactone degrading enzymes. Also for PET, we developed a method to look for the MS method to look for the derivative product. So in principle, we could use that for very high throughput to screen for new enzymes. So for DNA active enzymes, so we work with active enzymes because they were looking for mismatch nucleases. So we are designing now the substrate that we planned for the screening. So basically, for example, this is a mismatch with 8 nucleotides. So we have the fluorescence here and the potential here, and hopefully when the active enzyme is cleaved here, so this one should go away, and then you should have the fluorescence signal. Also, we also use the sequence mining to look for new nucleases, and we found one that is active. We have not tested with the mismatch, but we know that this is nucleases. So in another project on ancyloscope, so we look for silent debranching enzymes. So basically a couple of K-enzymes families that are known to degrade to debranch silent, and so we use the workflow also to look for those sequences, and so that is we are testing now whether we can use ECMAP-mapper. So it's also the way to annotate the protein based on orthologous group, and we found it's pretty useful because it can give you the overview of what kind of enzymes are there, and also together with the KEC reaction and also other information, for example, K-enzyme also. So we're trying to see now whether we can both use the profile HMM and also the ECMAP-mapper to find newer enzymes. So for functional screening, so we're developing the assay also for the silent debranching activities. So basically, we look for the turbidity, change in turbidity of the reaction. So on the opposite of the caprolactone, so when there is the silent debranching activity, so the media become more turbidity because silent become less soluble in the media. And so we're looking for the method using MS to look for the degradation products. So we are testing different methods now for the functional screening of silent debranching enzymes. So in another project, Aqua Health, we look for the microbiome associated with microalgae, and also the biofilm that is isolated from the water treatment. And basically in this project, we focus on looking for biofilm degradation enzymes and peptides, and also for antiviral activities. So we use the same enzymes mining method, looking for different type of enzymes that potentially degrade biofilm. And on the bioactive or antiviral activities, so we're using AntiSmart, and we're testing a program called Deep BGC. So basically, they use deep learning method to detect biosynthetic gene clusters. And for antiviral activities, so there's a new tool that was developed, I think, from Pasteur Institute, not mistaken. So they use a maxifier-based method that can look for antiviral gene cluster from a prokaryote that known to have actually antiphage activities. So we see if we can detect some cluster there in the data set. And for the last part, so also about enzymes, but in different contexts. So in the Marbles project, so we're looking for mostly on sponge associated actinobacteria. And we're interested in what is called tailoring enzymes. Sometimes it's called late-stage modification enzymes. So the enzymes, there's a wonderful reviews here about those type of enzymes. So those type of enzymes that makes a lot of decoration or some tailoring activities that create the diversity of the biosynthetic gene clusters. So we start with the MIBIC database. So it's a curated biosynthetic gene clusters with a lot of information. So we extracted information of the tailoring enzymes there. And then we also use ECNOV-Mapper that to annotate the BGCs from the MIBIC and to extract the relevant hits. And then, of course, we will come to the manual curations. So at the beginning, so we are at the beginning of this project. So we look for now the halogenase activity enzymes. So because it's, let's say it's a little bit easier to spot them. For example, here, you can see that there is the halogenase activity here that of these enzymes. And also in the marine microbes, so usually there's a lot of halogen bioactive natural products there. So just have the conclusion is that we found that in this silico data mining and in combination with the unique genetic materials, so there's a great potential there to find novel enzymes. And we saw that big data analysis, it has processed a lot, and then it gives you a lot of amount of data. But of course, the manual curation is always the bottleneck of the process. And wondering if there's a possible to automate also the decision making, whether you can have the shortlist automated also. And combination with the in silico data mining and functional screening, so you can find a very totally novel enzymes, because at the end, in silico data mining for now is still based on the homologs and something that known before. So functional screening can give you the enzymes that nobody have been seen before. Also, we learned that making all of the workflows a lot of work, but how to transfer that knowledge to your college, for example. So we're working with to apply the workflow language so that we can package the whole workflow into a single command line so anyone can run it. So I hope that I can give you some overview of the SynTef activities in the metagenome mining. So I would like to acknowledge my colleagues here at SynTef who participate in different projects and also our main collaborators in different projects here, and the funding agencies that have been provided us the funding to carry out those projects. So I brought 2 posters in to the conference. So if you wonder, so that we can discuss during the poster session. Thank you.
Thank you. So we will have time to discuss definitely these results and this new project at the poster session. But if you have any question, burning question now, you could ask. Do you have one? I have one for you. You're used to systematically use both SSN and multiple sequence alignments to define, to select your targets. How do you integrate or compare the results of these two different approaches?
I think a sequence symmetry network might be better because you have more or less the bird view of your sequence or your hits, and then you, in a way, you're able to select the candidate better. Because multiple sequence alignment, so you end up with with a very long list, and then it's a little bit tedious to look into them. So we use more and more sequence symmetry network, but at the starting point, multiple sequence alignment also work very well.
And what's the strategy to define the sequence similarity threshold finally in your SSNs?
Yes, so with the sequence symmetry network, we also play with different e-value because we observe in the project I have not mentioned here. So when we change the e-value, so that the network start changes, and then it will give you different conclusions on the novelty of your enzymes there. So I usually just use the one that
gives the best novelty.
People have used before.
To deliver something.
Yeah.
Okay. Thank you. Thank you. We will continue with the last. Sorry. Can you? I read the question. Any luck in finding efficient petases? There is a recent work on fast PTase that appears to degrade PT fully in 48 hours. Here is this paper. So did you succeed in finding new petases?
Maybe, but we no, we found some hits, but we need to confirm it, to confirm those hits. So we might have found some petase. Yes.
Okay. Thank you for this hope.
Thank you.
Plastic degradation. Thank you. Thank you again. And the last speaker of this session will be Adrian van Dijk from the University of Waterloo. Adrian is a PhD candidate, is it true? Yes, working on herbicide bioremediation. So maybe we could close the chat. Thank you. Please, Adrian.
So I'm in Trevor Charles' lab working on degradation of the herbicide glyphosate. Part of that is looking at our plans to, I'm working on making some metagenomic libraries, testing some metagenomic libraries, and on the path to that, it's important to understand how glyphosate interacts with both as a substrate, because it's got phosphorus in it, it can be used as a source of phosphorus in minimal media, as well as potentially a toxin to a number of bacteria. The pathway that targets, which we'll get into, is present in many bacteria. So in E. coli in particular, we want to look at sort of how that interaction is happening. A little bit of glyphosate to start. It's sprayed very widely. 180 million pounds applied per year in the United States alone. It's in Roundup, but it's also in various generics. It's been off patent for 40 years. And it's considered one of the safer herbicides that we have, and it's partially replaced much more toxic atrazine and acetylchlor. Atrazine is very toxic. It turns amphibians into hermaphrodites. We certainly want to reduce how much of that gets sprayed. And it can be used in place of tillage to kill the weeds rather than having to work at the soil. Working at the soil can often lead to a lot of degradation and a lot of soil loss. Estimated about 75 billion tons per year of soil loss. So 20 tons of soil for every ton of food we produce. Resistance to glyphosate is easy to engineer. It's been engineered into soy, corn, cotton, canola. And again, a lot of that is starting to come off patent. That was work done quite a long time ago. So we spray a lot of it, but it does generally degrade quickly, though it does sometimes degrade to aminomethylphosphonic acid. which is what happens when cut the bond right there. And the AMPA that's left, that is somewhat toxic. It's also phosphonate, so it's got a very strong carbon phosphorus bond. And that bond is very difficult to break down. So much of my work is trying to find enzymes that can actually break that down, look at the pathways that can break that down. And it targets the 5-enolpyruvyl shikimate 3-phosphate synthase enzyme, which is within the aromatic amino acid pathway. Aromatic amino acid pathway is found in plants, some bacteria, some fungus. It's not found in humans, not found in mammals or anything else, but it is responsible for 35% of the dry weight of plants through lignin. What glyphosate does is it interacts with the pathway right here, replaces phosphoenolpyruvate. You see, you've got some structural similarities and stops this reaction from going forward. So not all organisms that have got this shikimate pathway are killed by glyphosate. So there's a question of why is that? For one, the ZPSP, the target enzyme, EPSP synthase, comes in 2 versions, version 1 and version 2. Version 1 is generally sensitive to glyphosate, and it's always found in plants, found some fungus, found some bacteria. Turns out it's found in E. coli. However, single point mutation 2 EPSP synthase 1 can render it resistant to glyphosate, though it makes it much slower. So the organism certainly pays for that resistance if it doesn't need it. EPSP synthase 2 is basically always resistant. It's got actually a very similar active site, but doesn't have the catalytic inefficiency that EPSP synthase 1 has for that resistance. It's found some bacteria, including Agrobacterium, but it's not found in plants unless it's been engineered into them. Additionally, if an organism needs to use the shikimate pathway and is exposed to glyphosate, it can be resistant through glyphosate and glycine oxidoreductases. These are flavor proteins that cleave glyphosate right there. Again, creating that AMPA that we mentioned earlier that could be somewhat toxic. And that renders it, well, maybe more toxic to us. It is no longer toxic to the shikimate pathway. Or glyphosate acetyltransferases acetylate the nitrogen right there. So like I mentioned earlier, I'm looking at ways to break down glyphosate. That's most of the focus is in actually breaking it down. We can do that using a minimal media. Glyphosate has got that carbon phosphorus bond, that phosphonate bond. We can use the ability to break that bond as a selective means to find organisms able to fully break down glyphosate without having to worry about it creating AMPA, since it needs to break that bond. Normally, organisms do that through the CP lyase pathway, which is a fairly long pathway. This is not AMPA or glyphosate here, but it's a good example. And E. coli's got this pathway. It's got the canonical CP lyase pathway. So it's got all the genes we would actually expect it to be able to break down glyphosate. Our tests have been done using glyphosate as a sole phosphorus source, showing that it did not grow on glyphosate as the sole source of phosphorus. These were tests done by other people and published, and then we've also repeated these tests. So it was assumed something wrong with the CP lyase pathway. But then we did some more tests, and we looked at, you got to remember it's still affecting that shikimate pathway. So we wanted to test what happens if you just grow it in the presence of glyphosate. So we tested our minimal media. If you provide phosphoric acid, it's able to grow. The minimal media works, provides everything it needs. It's M9 based, only with the phosphorus taken out. But if you provide glyphosate and phosphoric acid, you no longer get growth. So E. coli is certainly sensitive to glyphosate. And then still can't break it down, but that's probably because it's being killed first. But it's still a question, if it were resistant, how would it interact? And we also did just a good control to show that the media is free of phosphorus, no added phosphonates. It's quite a while to actually get it sufficiently free. So again, this question still reigns, like, how will E. coli react when you provide it with glyphosate resistance genes, or what is missing to allow E. coli to grow on glyphosate? So one way we wanted to answer that was taking this condition and selecting a set of metagenomic libraries on glyphosate. Using E. coli HP101, we had a set of libraries from the Canadian Metamicrobiome Project. We've got these libraries in our lab. We screened these on that media with glyphosate as a sole source of phosphorus. The reason I wanted to use glyphosate as a sole source of phosphorus as opposed to just present in the media, since I'm working on breaking down glyphosate, I want to exclude anything that's just a glyphosate pump. I don't want to focus on things that can just pump the glyphosate out so it doesn't have to use it. And I want to know exactly what needs to be added to E. coli to get it to degrade glyphosate fully. Based on previous work, we would expect to find resistant EPSP synthases, resistant glyphosate oxidoreductases, possible resistant glycine oxidoreductases, and glyphosate acetyltransferases. And that's exactly what we found. We found through these screens, we found a handful of EPSP synthases, one type 1, which is almost certainly just a mutated type 1 that's resistant, and then 3 type 2s. These are always resistant glyphosate, so that's good. And then we found most of these clones came from sort of a wide variety of original organisms. We did this work using Phylopithia. Cosmos was sequenced by Illumina-MiSeq, and then we assembled it using Eugene Spade's algorithm. And these annotations were done by Proca and then pulled out manually. We also found, it's quite exciting, a handful of clones, 5 clones here that didn't seem to have any glyphosate degradation genes or any glyphosate resistance genes. One clone did seem to have 1289, did have a fragment of the CP lyase pathway, but not really enough to do anything with. And none of the rest of them do. So we think that's probably just coming along for the ride. Regardless, with these five cosmic clones, the next steps are to isolate the exact resistance genes. We hope there's something novel, of course. And then some of our collaborators over at Wilfrid Laurier University, just down the street from the University of Waterloo, they were interested in these glycine oxidoreductases. So they isolated 11AW19 and 11AW6. They didn't worry about 11AW8 since it's so similar. It's from the same organism, same group as 11AW19, and the sequence is very similar. And I wanted to see how does the activity of these glycine oxidoreductases compare to established glycine oxidoreductases in terms of resistance to glyphosate. So they cloned them into a pet vector and ran them in E. coli. They were able to confirm that their E. coli at very low levels of glyphosate is still inhibited. And adding the pet vector, adding the IPTG doesn't change that significantly. And what they compared this to is the canonical glycine oxidoreductase that provides resistance to glyphosate is from Bacillus subtilis. So they cloned that gene. And they showed, yes, it does in fact provide a little bit of resistance to glyphosate. And they compared our clones. They cloned the genes out of our cosmids. So these are isolated genes. And showed 11AW6 provides, again, a little bit of resistance to glyphosate, a little bit better than the glycine oxidoreductase from Bacillus subtilis. And then 11AW19 provides very good resistance to glyphosate. And in fact, they also compared this to glyphosate oxidase, which is one of the genes that's been engineered to break down glyphosate potential for industrial uses, to create resistant organisms. And we found that actually it grows as good or better than this engineered glyphosate oxidoreductase. So it's quite exciting that we've been able to use this screen to find really good glycine oxidoreductases better than some of the more engineered stuff that's out there. So certainly improvements we made by running these screens. So the next steps are to use subcloning to find those resistance genes in the cosmids I showed earlier that didn't seem to have anything annotated as a resistance gene. And then to create a CP lyase knockout E. coli so we can further look at the question of exactly how is E. coli responding to glyphosate. It looks like the story is almost certainly it's killed before it can break it down. But when we provide a resistance gene, then it's able to break down glyphosate. Any questions?
Yeah.
Hi, I'm just wondering if those clones that didn't have any glyphosate related annotations, if they had any other annotations.
Yeah, we ran Proca on them. So we got about 40,000 base pairs in the entire insert. We got back a number of genes, but none of them seemed to be related to glyphosate degradation. But we just tested that by looking for the known glyphosate resistance genes. So it made sure it, one screen we found a handful of different libraries. We found 5 that have nothing that we know about. There's almost certainly not 5 distinct, totally new resistance mechanisms. So when I screen that, I'll probably find the same thing that we already know about, but hopefully there's something new in there.
Okay, thank you.
Thank you. Any other question? Yeah.
Did you run the alignment of those 5 unknowns? Did you align those to make sure you didn't have duplicates? Because any duplicate in there would potentially be of unknown annotation, would be interesting.
Yeah. So they were aligned. They're not duplicates, but there's a fragment in 2 of them. There are, so I can explain this. There's a fragment in 2 of them that seems to be somewhat similar, but they're still annotated to totally different genes. But then when I look at those genes, they have nothing to do with glyphosate degradation or anything that we would sort of expect. So it's a possibility, or it may just be a coincidence. Thank you.
We have a question here in the chat. Did you use Proca with, oh, sorry. Did you use Proca with standard reference database or full UniProc KB?
I use a standard reference database.
Okay.
Sorry, I'm not a bioinformatician, but trying my best to run these programs.
Okay. Thank you. I have a question for you.
Yes.
Myself, did you try to detect the glyphosate degradation activity by testing the pH decrease, trying to detect the phosphoric acid release?
So we didn't look at glyphosate degradation that way, but that's certainly a good idea. We look for just growth or no growth. Our collaborators over at Wilfrid Laurier, they're more biochemists. They're hoping to do some of the actual glyphosate measurements to see it decreasing
Okay.
during the growth period.
Thank you.
Another one.
Just you look at the full database, you may get better.
Perfect. Yeah. So the full UniProc KB database. Thank you. Thank you very much.
Thank you.